Thank you. I need to start by thanking a number of collaborators that are co-authors on several of the papers that I'm going to present, and also not to forget many undergraduates and technicians who have done a lot of the dirty work in the field program that we have at Santa Barbara. I'm going to talk about the ecology of global change and infectious diseases, and I'm going to consider three aspects of global change. First, I'm going to talk about climate change. Then I'm going to talk about biodiversity loss, and then I'm going to talk about invasive species and how all those things may increase or in some cases decrease infectious diseases. Normally, when you read the popular literature and a lot of scientific papers, you see a focus on the increase in infectious diseases that are expected by some with global change. And I'm going to try to give a more complex set of predictions. So let's start with climate change. We know that a large number of infectious diseases, particularly vector-borne infectious diseases, may be or certainly are affected by climate. And we can tell that, for instance, by looking at the tropical distribution of diseases like malaria. And people have looked at these associations and built what are called process-based models, where you estimate, based on the current effect or association with climate, what malaria might do as the climate warms, and we get predictions like this, which are frankly quite terrifying if you live in Northern Europe or in North America. And papers like this have been used quite successfully to get the general public quite interested in the consequences of climate change for their personal lives. Well, the way that these models are built is we take what well-known aspects of the malaria life cycle, so for instance aspects of mosquito development rate or plasmodium development rate, and we go to the literature and ask how are those things affected by temperature. We build a model around those things, and that's how we can get things like a predicted association between temperature there on the bottom and transmission, which is here measured by something called R0. And the data, just to show you up in this upper panel, here's mosquito development rate as a function of temperature, and you can see that this is sort of expected as a linear increase. And one of the problems that I and others have criticized about these models is that we actually know, when we look at mosquitoes in the laboratory, that a linear increase with temperature for many of these rates is a gross oversimplification because the data actually look like this. And so in a working group at NCEAS led by my PhD student Aaron Mordecai, we asked a very simple question. What happens if we make these models a little bit more complicated to include these nonlinear effects of temperature on biological processes? Our approach was to go to the original data and fit curves to these data instead of straight lines, and then use the standard transmission models that others have used, and then use data from the field to ask which models better explain what we see in the field. And so here's an example of what the data look like. It's actually quite shocking how little data we have on the response of mosquitoes and plasmodium to temperature, but the data that we do have does suggest that those responses are nonlinear in all cases. We can then take these curves, put them into a transmission model that I mentioned, and generate, like other models have, a unimodal curve that relates temperature to transmission, but our curve looks quite a bit different from past curves relative to where the optimal temperature is for malaria transmission. Whereas past models suggested optimal transmission was at 31 degrees, our model suggests it's much cooler at around 25 degrees. This, of course, has big implications, but even more interesting is that no one had really noticed that the field data showed that temperature in Africa was occurring around 25 degrees, not 31 degrees. So what does this mean in terms of our estimates for where the climate is likely to be suitable for malaria transmission in Africa? We can take our blue curve there and apply it to temperature surfaces of Africa, and what you can see is on that scale bar indicates that there are areas of Africa where it is hotter than optimal for malaria transmission, and areas of Africa where it's colder than optimal for malaria transmission. And right in Central Africa, it's pretty good for malaria transmission, and that's consistent with aspects of transmission that we see in the field. So that's for March. We can do this for every month of the year because, of course, temperature changes with the season, and we can develop complex patterns of space and time for malaria transmission. If we sum all this information together, we can get sort of an overall index of how the area in Africa that's subjected to malaria transmission might change over time, and that corresponds to well-known seasonal patterns in some areas. If we just ask across all months of the year how intense is malaria transmission in Africa, we can generate a map like this where the darker colors indicate more intense transmission. And you can see again that Central Africa is much more suitable for malaria transmission than in Southern Africa or in the Sahel where it's too dry for transmission to occur. And then we can take this current estimate of climatic conditions for malaria and again expand them to projections of climate change and ask what is likely to happen. And we see, as you might expect, that there's going to be some changes, including, interestingly enough, that we expect to see reduced transmission of malaria in Central Africa, where presently malaria transmission is the most intense. I'll summarize these expected changes for you in the following bar graphs. This is the amount of area in Africa subjected to different types of malaria transmission, at least conditions for malaria transmission under different climate scenarios. And what you can see is a slight increase perhaps in overall area subjected to malaria transmission, but the most important aspect of this figure is that bottom set of dark bars which indicate that severe transmission conditions for malaria are going to decrease by half in 2080. And that's very different than what previous models have predicted that are based on these, I'd say, oversimplified linear estimates. And they expect that for all types of malaria transmission, there's likely to be a large increase. So the idea here is simply that by using slightly more complex models, we can get much better predictions, and they indicate quite different things about how malaria is going to change with climate. Now, since we're here in Sevilla, I'm going to use our models to make an additional point. This is a map of the number of months of malaria transmission throughout southern Spain and northern Africa. And what you can see is currently in Sevilla, we estimate conditions are favorable for falciparum malaria transmission about 6 months of the year. If we ask how that's going to change by 2080, what we see is that along the coast here in Spain, conditions are going to get better, more months of transmission during the year, but in the central part of southern Spain, it's going to become too warm for transmission, and conditions are going to degrade from malaria transmission. The reason I wanted to show this is that, of course, we don't have malaria transmission in Spain. We did, 50 or more years ago, but there have been a number of factors that have overridden the climate suitability for malaria transmission here and throughout North America and large parts of South America and parts of Europe as well. And so what are those things? Well, clearly, in developed countries, we have resources that we can commit to treating people with malaria. We can do mosquito control. And perhaps more importantly, we see big changes in lifestyle where people in developed countries drive in cars with the windows rolled up. We live in houses that have screens on our windows. We have air conditioning. And all of those things provide barriers between us and mosquitoes that reduce transmission. So there was a recent study, and the next figure I show you wasn't done by me, but I think it's very interesting. And this study asked, what are the joint effects of climate and economic development on projected increases or changes in malaria transmission in the future? And here's what they found, which was that there are really no cases where we should expect expansion of malaria in the future due to the overwhelming effect of economic growth on reducing malaria transmission. Hopefully that will be the case, and many of us will be around to see that, I presume. Okay. The next part of my talk is going to be about biodiversity loss and parasites. And there's a number of things that cause biodiversity to decline, and I wanted to mention that probably the most famous example of the relationship between biodiversity loss and infectious diseases comes from studies on Lyme disease. And there's been a proposed mechanism called the dilution effect, which suggests that there's a negative relationship between biodiversity and transmission of Lyme disease. And we've recently reviewed this, and even though this looks like a very complex model here for Lyme disease transmission, we've argued that it's far too simple. And in fact, when you consider other aspects of Lyme disease that are fairly well known, that it's much more difficult to predict the effect of biodiversity on Lyme disease. Now, Sarah Randolph gave a talk 2 days ago about this very subject, and so I won't go into a lot more detail about it, but I just wanted to point out and echo her conclusion that the dilution effect has been very overemphasized as a potential relationship between biodiversity and disease. Okay, well, what types of biodiversity loss are we expected to see? And some papers have expected that just the effect of climate change alone, we may see a decrease in species richness from 15 to 37%. What is that going to mean for parasite diversity? In our group, we build food webs, and we put parasites into those food webs. And here's an example of a food web from an estuary in California. And the red balls in this food web indicate free-living species like phytoplankton and fishes and invertebrates, and the yellow balls represent parasitic species. So firstly, you can see there are a lot of parasites in food webs. And secondly, when we have these model food webs, we can do experiments in our computer that simulate biodiversity loss. So for instance, we can cause a species to go extinct, and we can ask, are there any parasites in the system that are completely dependent on that host? And if so, they should suffer what we call a secondary extinction. And we can do these simulations thousands of times with different types of food webs, and what we find is overall there is a very simple and straightforward and actually expectable positive relationship between the diversity of free-living species and the diversity of parasitic species. Now, we can do these simulations in the computer, but we can also go out in the field and ask, do we see empirical relationships that are similar to this? And I wanted to show you an example using the bird community in our estuaries, the water birds that live there, and how those relate to parasites. So these birds are hosts for a community of trematodes that have a complex life cycle. And there's about a dozen trematode species that are pretty common in our estuaries. They all have a slightly different life cycle, but all of those trematodes go through the same first intermediate host snail. It's a mud snail that lives on the mud flats and the channel banks. And so we can go to a particular location, we can take a sample of 100 snails, for instance, dissect those snails, look at the parasite community in them, and in theory, the diversity of parasites in those snails should reflect the diversity of birds that are living in the surrounding area. So here's an example of one of our field sites. We can go to a tidal channel like this and easily collect a sample of snails and dissect them. Getting the information on the birds is a little bit more difficult. We use time-lapse videography, where we take a video for 5 seconds every minute for 12 hours a day over a tidal cycle, and then we have undergraduates who are very good at watching videos sort through the video clips and show us the ones that have birds in them. If the projectionist can click on this video, this is just some examples of birds moving through our area. We can identify all the birds to species very easily. We can tell whether the birds are feeding or whether they're just moving through the area. The videos, of course, work at high tide when it's difficult to normally sample. And then the video doesn't disturb birds, so we get a really good and accurate representation of what the bird community is doing when we're not there. So if we take these really good quality bird data and match them with the parasite data, what we see is a very strong and approximately linear association between the diversity of birds in a habitat and the diversity of parasites in a habitat. We get the same relationship also if we look at the abundance of parasites and the abundance of birds. So that's pretty consistent with our theoretical expectations, but I wanted to point out that food webs are more complicated than that. And so not only do we see secondary extinctions happening in food webs, but there's the potential for a lot of indirect effects. I'm sorry, I skipped ahead of myself. I wanted to ask, under the scenarios of climate change, we're expecting 15 to 37% biodiversity loss. What does that mean for expected parasite diversity loss? Here's that, what we expect we're going to happen in 50 to 80 years for free-living diversity, and that suggests that there's going to be a subsequent loss in parasite extinctions due to these secondary extinctions. But parasites might also be subject to primary extinctions when they fall outside of their thermal optimum when climate changes. And so we might expect even additional parasite extinctions. On top of that, the overall prediction is it's quite possible that parasite diversity loss is going to happen much faster than free-living diversity loss. Not that anybody really cares, frankly, but that's the predicted pattern. Okay, back to the food web. As I was alluding to, the food web is more complicated. There's additional things besides secondary extinctions. We have potential for indirect effects, and I'll give you another example. So imagine we have an extinction of a particular species. We might expect that the prey of that species could increase in abundance, and that the parasites of that prey could then increase in abundance. So there's an example how extinction could lead to an increase in a parasitic species. And I'll give you an example for where that has importance for human diseases. So we're particularly interested in a disease called human schistosomiasis. It's a disease caused by a trematode. It's a snail vector disease. And one of the, it occurs throughout tropical areas, but particularly in Africa. And the epicenter recently for schistosomiasis transmission has been in the Senegal River. Schistosomiasis wasn't always common on the Senegal River prior to the 1980s. In fact, it was relatively rare. But the World Bank funded a major development project in Senegal called the Diama Dam, and the dam blocked the tide from going upstream so they could grow massive plantations of sugarcane. Now, we know that dams have a lot of effects on aquatic food webs, and we've been interested in asking, what is it about this dam that might have changed the ecology? Because one of the things that we know changed due to the dam is a huge increase in snail populations and schistosomiasis transmission, leading to the highest epicenter and examples of schistosomiasis transmission in the world. One of the species that used to be common upstream was a giant river prawn, and the fishermen used to catch this prawn and take it to market. It's now extirpated from the Senegal River. And we've known from past work that we've done that prawns love to eat snails that are hosts for schistosomiasis. So we can build microcosms in our lab and watch snail populations grow and then add prawns to these microcosms. And here's an example of the type of data that we get. Without prawns, the snails increase in number until they reach some sort of carrying capacity, but when we add the prawns, the snail populations are invariably extirpated with time. So this has led to a prediction that if we can restore prawns, if we can restore the food web of the Senegal River, we may be able to bring the system back into balance, and that might have positive health effects. We're doing that in the field as we speak. There's a multinational project called Projet Crevet, and this is actually one of our experiments. We put in a large cage in the area where local villagers swim and do their laundry and get their drinking water. And then we go and we treat all the individuals in the village with praziquantel, and that cures them of schistosomiasis. And then in half of those areas, we introduce the giant river prawn, and at the moment we're trying to see whether or not reinfection rates of the local villagers are slower in the areas where the river prawns have been introduced. And so far, that is the indication from the preliminary data that reinfection rates are very low where we've reintroduced the river prawns. Oh, gosh, okay. So that's one disease, one human infectious disease. What about the rest of them? We've been working on a tabulation of all the protozoan and metazoan human infectious diseases that are relatively common and cause some pathology in humans, and asking this question: should we expect to see an increase in disease with biodiversity or a decrease in infectious disease with biodiversity. And so what we've done is for each of these infectious diseases, we've looked at their life cycle. We've asked, are they vector transmitted? Do they have an alternate host? Is that alternate host a rodent or a primate or a carnivore? Are these infectious diseases associated with intact forests or not, et cetera, et cetera? And from this information, we've constructed a set of logical criteria by which we can generate a list of hypotheses for each parasite about how it will respond to biodiversity. And I just wanted to show you a tabulation. This paper is just accepted in ecology that I think gives us a view of what we might expect in the future. So the first thing is that of those 69 infectious diseases, quite a lot of them are probably not strongly related to biodiversity. These are infectious diseases that transmit directly from person to person. They don't have a vector. They don't go through an alternate animal host. And so we don't expect them to change much with biodiversity. There are examples, like the human schistosomiasis case that I just gave you, where infectious diseases are negatively related to biodiversity. Higher biodiversity will presumably protect people from infectious disease. But far more common are examples of infectious diseases that are positively associated with biodiversity, about 3 times more common. So what this just simply shows is that the general message that conservation biologists have given to the public has been that biodiversity is going to protect them from infectious diseases. And that may occur in some cases, but I don't think it's the overall trend. Well, the third part of the talk is about invasions and parasites. And this is a photograph of my colleague, Mark Torchin. And in his right hand, he's holding a very large crab from Europe. This is the European green crab. And in his left hand, he's showing a normal-sized crab from San Francisco Bay, where the crab has invaded. You can see that the invaded crab is much larger, doing much better in San Francisco than where the crab is native in Europe. And so we've been interested in going to regions around the world where the crab is native and where the crab has been introduced. The crab goes from Norway down to Gibraltar, and then there's 6 different places around the world where it has invaded. We go to these areas, we estimate their density, we look at the sizes of the crabs, we dissect them for parasites, and in Europe we find a large number of parasites. They're very prevalent, and have a large number of groups of parasites. And then comparatively, when we've gone around the world to these other locations, we find that parasitism is either absent or much reduced, and in general of much lower diversity. So this suggests that this crab has been released from parasites as it's invaded. And to ask whether this is a general phenomenon, we did a literature review of 26 animal taxa where there are good data from introduced and invasive populations on parasites. And what these data show is that on average, where an animal is native, it has about 16 reported parasites. But in introduced populations, it only brings 2 of those parasite species with it. Introduced species pick up an additional 3 or 4 parasites, so overall, introduced species tend to have about half the parasite species compared to where they're native. Now, there's a couple of implications of this. The first is that this may result in increased performance of invasive species, a concept that's been called enemy release. But also from another perspective, the fact that every introduced species is bringing 2 parasite species with it means that the invasion problem is much bigger than we had originally thought. And I'll give you an example of how that can affect food webs. We've been working in Norway with some Norwegian colleagues on putting together a food web for an Arctic lake, Takvatn. And this food web is represented here, and as we do in other systems, we look both at the free-living species, which are indicated by the black lines, predator-prey relationships, and the parasite species, which are indicated by the red lines, which indicate host-parasite relationships. And in this lake, there were 2 species introductions, and those species brought new parasites with them, so that you can see on the right-hand column there, there are 5 additional parasites that come in with those invaders, and they have completely changed the structure of this food web, making it much more complex than originally thought. Well, I'd like to make 3 conclusions. The first is that climate change can have fairly complicated effects on infectious diseases. In general, the prediction is that it should shift the climate conditions for all species, infectious diseases, free-living species, it doesn't really matter. Not necessarily will climate lead to an expansion of infectious diseases. In addition, while climate suitability is expanding shifting for infectious diseases, that doesn't necessarily mean that we're going to see increases or changes or shifts in transmission, because there are other covariates, particularly economics for human diseases, that will override climatic effects. In short, I think that the best hypothesis we have is that we're going to see decreases in infectious diseases with global change overall, not increases. Secondly, biodiversity loss, counter to most claims that you'll read in the literature, mostly reduces parasites. There are cases where it may increase parasitism, but mostly it's going to reduce parasites. And then finally, invasives both bring and escape parasites, and both of those things are bad news. New parasites means new host-parasite relationships that are very difficult to predict and can cause problems and have been shown to cause problems. And then the escape of parasites by introduced species may increase their performance and increase the problems that they create in natural systems. Thank you very much.